So I already talked about a lot of this on the previous slide, but here we can kind of see where we must have a virus-infected cell. So it has taken an antigen, processed that antigen, and then placed a portion of the virus as antigen on the MHC class I receptor. So when a T cell recognizes it, this T cell is going to become activated and differentiate into cytotoxic T cells. So these cytotoxic T cells are going to circulate through the body, looking for any cells that have that same antigen in their MHC class I, because that means that they are infected with that virus. And they are going to release chemicals that are ultimately going to kill the virus-infected cells. They can also do the same type of mechanism for cells that are cancerous as well. It is important to note that memory cells will be generated as well in the event that you get infected with the same virus again in the future. They can start circulating right away to get rid of any potential virus-infected cells before you actively feel sick. We do have natural killer cells as well, which can also kill virus and cancer-infected cells. However, they work very similar to cytotoxic T cells in the sense that they release chemicals to kill virus and cancer-infected cells. But the main difference between a cytotoxic T cell and a natural killer cell is that natural killer cells are going to kill non-specifically, meaning they are able to kill any cell that's infected with any type of virus or potentially has become cancerous. So, for example, if you became very unlucky and infected with COVID-19, measles, and the flu. So you have cells in your body that are infected with those different viruses. You would need to activate 3 cytotoxic T cell lines in order to fight that infection. So you would have one type of cytotoxic T cell that would go around and kill any of the cells that were infected with measles. You'd have another going around killing any of the influenza-infected cells, and then a third going around killing any of the COVID-19 infected cells. But with natural killer cells, one natural killer cell can go around and kill all 3 different types of virus-infected cells. So they can do it non-specifically and just really wipe out any cell that's infected with any type of virus. The downside to them is they can sometimes harm healthy cells, so there can be more side effects, whereas cytotoxic T cells, they can kill virus-infected cells with better precision. The next lymphocyte are B cells. So B cells, they mature in the bone marrow, hence the name B cell. And when a B cell becomes activated, and again, that's typically in response to interacting with a helper T cell, but when a B cell becomes activated, it is going to differentiate into plasma cells and memory B cells. Plasma cells are ultimately going to produce antibodies that are then going to go bind to antigens on the foreign pathogen and mark them for destruction. The memory B cells are going to circulate in your body throughout the future, and then should you get infected with the same pathogen again, they're going to differentiate right away into plasma cells and produce antibodies to fight off that infection before you actively get sick. So with B cells, they're very important in their ability to differentiate into plasma cells, which ultimately produce antibodies to fight off the infection, and then memory cells, memory B cells, which give you that long-term exposure or protection towards exposures in the future. So we know that plasma cells produce antibodies. So what do these antibodies actually do? Really, antibodies are going to bind to antigens on foreign pathogens and essentially call for their elimination from the body, so it's going to help cure infections. So when antibodies bind to antigens, they work kind of 4 different mechanisms One being opsonization. Opsonization is just enhanced phagocytosis. So if we have a pathogen in our body and it has a bunch of antibodies bound to antigens on its surface, it is going to call for phagocytic cells to arrive on site much faster to phagocytose and eliminate that pathogen. So opsonization is just enhancing phagocytosis by letting the phagocytic cells, like the macrophages, see the foreign pathogens much more easily because they're kind of all marked with antibodies at that point. So it just allows for a easier route of recognition by phagocytic cells. Then there's neutralization. Neutralization is the coating of antigens on the surface of the pathogen with antibodies to prevent it from binding to its target site. So this is very helpful with viral infections. If we remember, viruses have spikes that they use to attach to receptors on our cells. And what antibodies can do is they can bind to the spikes on the virus and prevent it from being able to attach to receptors on our cells and infect our cells. So essentially, they neutralize the effect of the virus. They can also bind to toxins produced by bacteria and neutralize the effect of the toxins that could potentially cause things like muscle spasms or paralysis. Then there's agglutination. Agglutination is the ability for the antibodies to bind to several bacteria or viruses at the same time. So if you look at antibodies, they're shaped like a Y, and at the top of that Y, they have two binding sites. So that means they can kind of hold on to two bacteria at the same time. So when phagocytosis occurs, a lot of times the phagocytic cell is just kind of engulfing one bacteria at a time, so it may take a little while. But if antibodies are present, what they can do is kind of grab hold of multiple bacteria, the antigens on multiple bacterial cells, and kind of lock them in so the phagocytic cells can come along and engulf numerous bacteria at the same time. So it just kind of speeds up that phagocytic process. Another thing, when antibodies are present, specifically in the blood because you have an infection, this can call for the activation of complement, which we talked about in the last lesson. If we remember, complement is a series of proteins in the blood that when they become activated by something like antibody production, they will form an MAC complex or a membrane attack complex, which is a ring of proteins that will sit on the surface of bacteria, which we see here, and essentially poke holes in the cell membrane of the bacteria, which causes it basically to shrivel up and die. It's kind of like they're stabbing the bacterial cells. So antibodies are very important, and they are one of the key factors to overcoming an infection. And then also with the memory cells, their production much quicker can prevent us from getting secondary infections in the future due to secondary exposure. So we need to talk about primary and secondary response, immune response. So the primary response is the first time you get sick with an infectious pathogen. So this would be, the first time you get measles or the first time you get influenza. The very first time you get sick, it takes a little while for your body to activate the appropriate cell lines. Because remember, we have to find the right T cells and the right B cells and go through all those activation pathways. But over time, usually about a week or two, the cell lines become activated, and then our body produces antibodies to fight off the infection, and then ultimately we get better. So that first time you're exposed, you're going to get sick, unfortunately, but your body's going to fight back. It's going to produce antibodies, which we see here, and then you'll get better. So let's say a few months later, you get sick with the exact same pathogen all over again. Well, as soon as that pathogen gets in your body, those memory cells, like the memory B cells, they are going to recognize it right away, And without any of this lag time that we had before, those memory B cells are going to differentiate into plasma cells and start producing massive amounts of antibodies instantly. So those are going to be expelled into the body, and they're going to bind to the antigens on that pathogen right away and eliminate it before you even know that you were sick. So in that secondary response, the memory response allows antibodies to be produced much faster and in much higher quantities so that you are able to clear that infection before the infection really takes hold and is able to cause signs and symptoms. So you likely will not even know that you're infected, know that you've gotten sick that second time. Now, viruses and bacteria have a way to overcome this process. They can mutate. And unfortunately, if they mutate, they change themselves enough that any memory cells that you have may not recognize them. And if they don't recognize them, they're not going to differentiate and produce those antibodies to fight it off. And then unfortunately, you are going to get sick that second time. I mean, and your body would activate the appropriate cell lines and produce new antibodies to fight off the infection, but if it mutates, you are likely to get sick again. But hopefully it doesn't mutate, and those memory cells recognize it, and you produce those antibodies much quicker and clear the infection from your system.